Biochemistry Module Lecture 1, Amino Acids, Peptides, and Proteins. We'll start off by talking about what exactly amino acids are. Put simply, they are the fundamental building blocks of proteins, and they're comprised of an amino group, a carboxylic acid group, and an R group attached to a central alpha carbon. Note that this R group, or the side chain as it's commonly called, is going to dictate the function. There are hydrophobic and hydrophilic amino acids, and this is based entirely on the polarity of this R group. Also take note that there's 20 variants of this that you will see in eukaryotes. When you look at it from a stereochemistry standpoint, there's only two configurations of amino acids. There's D and L, and this is based on where the amino and carboxylic acid groups are with respect to each other. All amino acids are noted to have an S configuration except for cysteine, and they're all obviously chiral except for glycine, which has just a hydrogen as its R group. Eukaryotic amino acids are all L, whereas D amino acids can exist in prokaryotes. This is largely based on how we make proteins within eukaryotic cells. Below is a list of the side chains and a way to categorize them. Note that all amino acids have a 3-letter abbreviation and a single-letter abbreviation. And it's really important that you have these memorized. On the MCAT, you're going to see these in sequence based on these abbreviations. So you need to know how to recognize these and what they are. The way I like to divide them is by nonpolar, non-aromatic, and the ones are listed below. Aromatic, polar, and acidic and basic are especially important when we talk about things such as PI, which we'll go into later. Below is the 20 amino acids you are going to see, and the R groups. It's important that you have these R groups memorized and what these amino acids are called, because when it comes time to take your test, you're going to see just the abbreviations, and you're going to have to know what kind of characteristics each of these are going to have. This is especially true when you're looking to see if a certain protein is going to exist within the membrane, extracellularly, or in hydrophobic areas. Having a good idea of what the structure is of these amino acids is going to help you discern these things on test day. When we talk about amino acids, we have to look at the acid-base chemistry. Amino acids are noted as amphoteric, and they're also zwitterions. If you remember, amphoteric means that it can exhibit acidic and basic qualities. A zwitterion means that it can have a positive and a negative charge. Usually that goes hand in hand with amphotericity. Remember that pKa is noted as the pH at which half the species is deprotonated, and the concentration equivalent is listed below. The form of the amino acid is going to depend on the pH. When the pH is low, it's going to be fully protonated, which makes sense because at low pHs, there's a lot of H+ floating around. If the pH is near the Pi, that means that it's going to be a neutral zwitterion, and at high pH, it's going to be fully deprotonated, which would make sense if there's lots of hydroxide floating around. There are amino acid titration curves, but they're very similar to other titrations. Note that the curve is almost flat at the pKa values, and it's nearly vertical at the Pi value. This is probably approximately where you're going to see the equivalence point in a normal titration curve. When you look, the amino acids have two groups in particular that you have to think about. So I'll draw a slight simplification here, but with an R group, you're going to see that one side's going to have the carboxylic acid, and the other side is going to have the amino group, like so. What you're going to notice is that this hydrogen can come off, and this hydrogen can come off as well. And as they add, it's going to change. So for this one right here, this is going to have a positive, but upon removal, we're going to have a negative, or a neutral here, and upon removal here, we're going to have a negative, which is why we say that there can be two charges noted. This can either have a positive amino or a negative carboxylic acid group. So just take note of that. We mentioned Pi, and Pi is listed as the pH at which a particular molecule will carry no net charge, and the equation is listed below. It's simply the pKa at which you're going to see ammonium positive versus the pKa of the carboxylic acid being protonated, divided by 2, because it's an average. If it's a charged R group, you're going to have a Pi around 6, and an acidic is going to have a Pi of substantially less than that. Basic, obviously going to be substantially greater. This is based on the fact that the internal environment of most organic life is going to be slightly acidic. Now that we've talked about the fundamental building blocks, let's go into proteins in general. Proteins are formed when amino acids exhibit peptide bonding. These are bonds that are formed by condensation or dehydration reactions. Amino groups of one amino acid is going to link to the carbonyl group of another. These amide bonds are rigid, so the proteins that are formed from this are going to have to fold in other ways. These bonds don't rotate very much. Breaking these bonds can be done via hydrolysis. The cell is going to use condensation reactions and hydrolysis to regulate these proteins accordingly. Below is a diagram listing how these bonds form. As you can see, the OH from the carboxylic acid of one and the amino group on the other are going to condense out, meaning that water is going to form from this formation. If we were to add water and use it to hydrolyze, then you would simply see it reverting back to the original. For the last bit of the lecture, we're going to go into protein structure, and there's 4 levels of it. Primary structure, to start off, is the linear sequence of these amino acids, and it's the components of a protein. So if you look on test day, you're going to see listings of primary structures like so. And you're going to have to know what they are just by looking at these sorts of abbreviations. As they're listed out, generally you're going to see characteristic trends, so be aware of them. The next level we're going to go into is the secondary structure, which is noted as the local structure of neighboring amino acids. These are generally because of hydrogen bonding, and there's certain amino acids that are going to disrupt this, starting with proline. Proline is cyclic, so it's going to have a tough time doing this. And there's 2 particular secondary structures that you're going to observe. They are alpha helices, which, as it sounds, is helical surrounding a central axis, and beta sheets, which are rippled as parallel or anti-parallel depending on structure. A diagram of how these structures look is listed below. Note that these are based on hydrogen bonding, so they're non-covalent. Tertiary structure, on the other hand, is based on covalent interactions. They're 3D structures that are stabilized by hydrophobic interactions, acid-base interactions, hydrogen bonding, and disulfide bonds. Disulfide bonds being covalent. The hydrophobic interactions are when R groups that are hydrophobic are pushed to the interior of a protein. This will increase the entropy of the surrounding water molecules, thus decreasing the delta G. Disulfide bonds are between 2 cysteine residues. If you see cysteine residues, you should immediately be thinking about disulfide bonding. A common hang-up with students is, what is the distinction between secondary and tertiary structure? Secondary is noted as local structures, whereas tertiary can be across the molecule. As you can see in the bottom right picture, the cysteine residues that are exhibiting disulfide bonds are actually pretty far away from each other, but they can still bond with each other to form the tertiary structure. Another separation is that tertiary structures can have covalent interactions, whereas secondaries cannot. Make sure you have this distinction clear when it comes time to take your test. The final level of structure that we need to go into is the quaternary structure. Note that not all proteins are going to have quaternary structures, but when you see complex formations, it's almost always going to have some sort of quaternary interaction. These are conjugated proteins, and they're generally going to covalently attach to molecules such as metal ions, vitamins, lipids, and carbohydrates. The most common example you're going to see on test day is hemoglobin, which is made by 4 heme groups around iron residues. Below is a diagram of what this is going to look like. If you remember in our biology unit, we talked about how hemoglobin functions and how these proteins are going to cooperatively bind with each other, and how they're going to cooperatively bind with oxygens. The final thing we'll go into is what denaturation is. This is the loss of the 3D structure that's due to heat or solute concentrations. This is going to result in the loss of protein function. When we talk about proteins, we have to remember that their function is directly tied to their structure. So the loss of this structure is going to immediately result in the subsequent loss of function. We'll finish our first lecture in this series there. Please join us in the next lecture as we go into probably one of the most important proteins in the biological world, enzymes.